Hello guys, welcome again to my YouTube channel. Today I'm trying to deal with Sysmex-SP line. So we've already talked about something like Sysmex-SP full block count. So we've looked at how the reagent work. So now we're going to look at something again that is different. After the full block count analyzer, the next thing you then see is the SP10 that can automatically make the slide. So how do they work? Which reagent do they use? So basically they use a form of Romanowski stent. Remember that Romanowski stents, they are kind of multi-stents, okay, where they contain both the acidic stent and also acidic dye and basic dye, okay. Now Romanowski stent, you can get something like Jimsa, you can get something like Leishman, you can get something like May-Grunwald. So but when you look at the Sysmex, what they normally use in Sysmex, what the Sysmex analyzer use is Jimsa, then it's also used May-Grunwald stent, okay. So like I've said, so these stents, they are neutral stents where they contain both the acidic dye and also the basic dye, okay. But this is the two stents that is commonly used. And of course, again, another thing it uses is methanol. And what does methanol do? They use the methanol, one, to fix because after the film is made, you allow it to dry. Follow the dry, then you fix it. So why are you fixing this slide? You are fixing it because you want everything that is on that very film to remain intact. That will then help once it's turned, you see. Because if you don't fix it, something can contaminate it, or maybe something that is there, may be affected by many factors, okay? So because of that, you don't want that. That is why you fix. You fix to keep everything on that very film intact, okay? So that's why we fix, okay? Now, following the fixation, so then the stenting can now take place. So that's why you fix. So I've mentioned that with the SP10, they use May-Grunwald, they use Jimsa, then they use, of course, methanol, which is for fixation, okay? Another thing they use is phosphate buffer. So what does phosphate buffer do? Phosphate buffer is used for the preparation of the blood film. Nothing that the SP10 uses. SP10 uses cell pack, okay? Now let's look at it in detail how these things work. So remember that once the blood passes to the SP10 will aspirate the blood, okay? Once it aspirates, that means that channel then will contain blood. So but with the help of the cell pack, which is a diluent or normal saline, what would then do that it will then flush up that place. It kind of used to rinse that very place to make sure that it is free from any blood stain, following that aspiration. So that is what the cell pack is doing. The May-Grunwald stent is used for the staining of the blood, okay? And of course, the Jimsa is also used for staining of the blood. Remember that I've told you that these stents, they are neutral stents. They can stain both the acidic part of the cells and also the basic part of the cell. Remember that when we talk about the acidic part of the cells, you might be looking at something like the nucleus, and then when you talk about something like the basic part of the cell, you're looking at something like cytoplasm, okay? So something like that is what you're looking at. So anyway, they stain both of them. So depending on, from the staining of both of them, that will not give you a clear picture of what you are looking for on the cell. In other words, it will expose the red cell very well for you. It will expose the white cell very well for you. Everything that is on that very blood, it will expose it very well for you. They expose the idea that because they are exposing it very well for you, it cannot help you to see all the features that may be either normal or abnormal. So methanol, like I've said, it can be used for to fix the slide after it's been made. Following the drying of the once you make the film, it dry, you allow it to dry. So analyzer will allow it to dry. In fact, there's something I need to even say before I go ahead with it. So once the sample goes into the analyzer, then it will aspirate the blood, mix it, okay, then aspirate the blood. Once it aspirates it has a slide where it can use to make the film. Once it makes it, then it has the inbuilt fan, okay, that will fan it to help it to dry. Then following the drying, then it can then push it, okay, to now be fixed, okay, it will be in a rack, push it to now be fixed. Then after the fixation, then it can now take it to the May-Grunwald and also the Jimsa to be stent, or right stent as it gets to stent it. Then following the stent, the stent will then help you to see the clear picture of the cell content, then allowing you to be able to identify whatever thing that may be abnormal or even normal, okay? Then we'll look at methanol. In addition to the fixation, methanol is also used in the maintenance procedure. So yeah, that is for SP10. Once again, with the SP10, it makes the automated slide. So once it makes that very automated slide, then it allows it to dry. Following the drying, then take it to the methanol, fix it, and following the fixing, it can then be stained. And once it's stained, then it can bring it out for you, and of course allow it to dry. Then it now allows you to be able to see whatever thing that you want to see. But what I want you to get here is that with the SP10, it uses Jimsa, it uses May-Grunwald, and also some of them are used rice stem, okay? Then you have something like methanol. So you now know the functions of each one of them. I'm trying to tell you that both May-Grunwald both gypsum, okay, they are all form of Romanesque stem, which that can be able to extend the acidic content of the cell and also the basic content of the cell. So here you go with the SP10. These are the reagents that they use, and this is the function of those reagents. Then, if of course, if they ask you the principle of SP10, now you now know how it functions, what that it uses, and all of that. So what I'm going to say that if you do have any question, please could you put it in the comment section and let me know any question you have, or also how the video has also helped you. And please, can I also ask you to like, subscribe, and share. Thank you very much till I come back away again. Bye bye.